Our next panel is entitled Of Mice and Men, Cancer in the Workplace. The moderator is Dr. Umberto Saffiotti. He was born in Milan, Italy, is now a resident of Bethesda, Maryland. He's a participant on the cancer panel and will be discussing carcinogens, their nature and identification. Dr. Saffiotti has been with the National Cancer Institute since 1968 and currently is chief of the Laboratory of Experimental Pathology in the Carcinogenesis intramural program in the Division of Cancer Cause and Prevention. From 1968 to 1976, he served as the Associate Director for Carcinogenesis of the National Cancer Institute. Dr. Saffiotti.
Thank you. I'm concerned that I have been asked to serve as the moderator or chairman of this session, and we'll have to try and fit a long program in a very tight schedule. We are already somewhat over time. So the plan is to have the first three talks given in your program, about 15 minutes each. We will have to try and be, if possible, even shorter because we are running out of time. We'll have a short coffee break. And then after that, there are 3 rather than 2 talks. Dr. Olson has been added to the program between the talk by Dr. Epstein and that from Dr. McCarville. So we have a very heavy schedule. I want to try, if we can, to leave some 10 minutes at the end for discussion, and therefore we will proceed fairly rapidly throughout it. The first speaker of the panel is Dr. Thomas Mancuso from the Graduate School of Public Health of the University of Pittsburgh. Dr. Mancuso has a long and very distinguished history of research in the area of occupational epidemiology and occupational cancer, and I think his work is known to most of you. He doesn't need any further introduction, and we'll be glad to hear him talk to give us an overview of this complex problem. Dr. Mancuso. Identifying carcinogens relates to what we know about their nature because we have to develop appropriate methods for the detection of their biological effects. The problems of epidemiology have been already outlined by Dr. Mancuso, and in previous discussions at this meeting, Dr. Roll has very appropriately indicated the difficulty of utilizing a method that relies on the manifestation of a long-term effect in a population as a means to protect that population from potential hazards. Simply stated, we cannot wait for human cancer evidence to accumulate to an extent that it can become detectable epidemiologically before we can intervene with some protective actions if we have adequate evidence of other kinds. The other kinds of evidence that have been widely used for this purpose are of an experimental nature. Now, you have heard over the years a lot of discussion, some of this sort of ironical, about cancer in rats not being relevant to cancer in man or all such things. Let me just pause for a second and remind you that the progress in medicine in the last century is largely based on what we have been able to see in our laboratory animals. The development of experimental medicine as a major tool, as a major way to lead towards the progress of scientific medicine that started in the middle of the last century has been very largely based on the careful and controlled, I repeat, controlled reproduction of disease patterns in animals where you can actually have a controlled experiment and see what factors are responsible for certain agents, for certain effects, and which ones are not. So the reliance on experimental animals is a time-honored basic tool in medical research. Obviously, one has to make sure that we have animal models that are relevant, that are similar to the human type of disease that we want to study. And here is the important point that over the last several years, but particularly in this past decade, most major forms of human cancer, as we know them from human pathology, have been reproduced in experimental animals by chemical induction. that is, by treating the animals with chemicals, you can induce in these animals most of the major types of cancers that are known to be representative of human cancer pathology. Now, that is, for example, the case with cancer of the respiratory tract, cancer of the larynx, cancer of the bronchus, which is the major type of lung cancer, in their various histologic types. It is true for cancer of the digestive tract, cancer of the esophagus, which is a very prominent form of cancer in parts of the Asian continent. Cancer of the stomach, which is somewhat it's declining in the United States and some other countries, but still of a fairly high incidence, is not to be dismissed. And very important, cancer of the large intestine, which is in fact increasing in the United States and several other countries. All these are reproducible in animals by chemical induction with a pattern very similar to that seen in the human. Cancer of the pancreas, which has been, for which animal models have been developed in the last few years, which is a major form of cancer in the human, and one that again is on the increase. Cancer of the liver, which is not a very high frequency in the United States, although there are some suggestions that it may be increasing, but it is a very high frequency cancer in other parts of the world. Cancer of the kidney, cancer of the urinary bladder, cancer of the mammary gland, and so on. Not only can we see the reproduction of this pathologic entity, but we can see that the steps towards the development of these cancers, the cells of origin, the early changes, are becoming more and more qualitatively recognized as being very similar in the animal models and the human counterpart by studies that compare the two. We are now much more informed, say in the last decade, about the mode of action of chemical carcinogen, their fate in body tissues, the way they are changed metabolically to become reactive molecules that interact with target biological molecules in the cells that are hit by these chemicals. We know much more about the specific chemical interactions of chemical carcinogens with the target macromolecules, and these includes the genetic material, DNA, the messenger and RNA material, and the proteins. Now, we are, in the last few years, we know in the last few years from work in several laboratories that there are specific components, the bases in nucleic acids, that are hit by carcinogens in specific ways, so that we have been able to identify the specific interaction products, chemical products, of carcinogen and DNA bases that have a considerable degree of chemical specificity. And there are now very sensitive methods that have been developed, including some that were recently developed by Dr. Harris in our laboratory, to identify extremely small quantities of these interaction products in the tissues. So we are beginning to see how to go about mapping the fate of these chemicals in the body, which target cells they hit, where do they bind preferentially, and what happens subsequently. All this has now been studied in comparison in animal and human tissues. Now, how do we study the effect of carcinogens in human tissues when obviously we cannot make experiments giving carcinogens to people, because it would be obviously unethical and hazardous to do so. In the last few years in our laboratory, the work of Kurt Harris and colleagues and work in other laboratories has shown methods for the culture of human tissue explants in vitro. That is, you take, for example, from surgical material, a piece of normal bronchus or intestinal mucosa or esophageal mucosa or pancreatic duct, the target organs of cancer induction. You can then take these tissues and maintain them in appropriate culture conditions in the laboratory. They become experimental tools. You can then put carcinogens on those human tissues. You can do the same with the corresponding animal tissues. This line of work has been very exciting in the last few years, 2-3 years that this has been established, because it has allowed us to show the correlation between the pattern of reaction of animal tissues and the corresponding human tissues to carcinogens. The message that has come out of this is that there is once again a very striking qualitative similarity, all the way to the molecular manner of interaction of chemicals forming specific target reactions with the genetic material of the target cells. However, in all these studies, as well as in long-term studies of tumor induction, one can see considerable variations on the quantitative level. For example, species to species differences that are very marked quantitative differences. In fact, some species or strains of animals will be very resistant to certain carcinogens, others will be very susceptible, some will react by producing tumors at certain sites in certain organs, others in other organs, so that it becomes very difficult to predict the specific reaction or the specific response quantitatively from one set of conditions to another. In the work on the interaction of carcinogen with human tissues, another factor is coming out very clearly, which shows a marked inter-individual variation in the quantity of carcinogen that is bound to these target macromolecules in the cells. That is, my bronchus can bind a certain amount of, say, benzpyrene in the air. Somebody else's bronchus may bind 100 times less. In that case, maybe I may be 100 times more susceptible. We don't know yet enough to be able to correlate precisely these potential indices of exposure at the tissue level with susceptibility. But they are an encouraging development in giving us potential markers for susceptibility studies. Now, the development of in vitro methods, that is, methods in which cells or tissues are kept in culture conditions in an incubator and can be treated with carcinogens, have been a very valuable tool added to the arsenal of methods for the study of the effect of carcinogens in the last few years. And they include effects on in the induction of mutations. They include effects on the transformation of normal cells into cancer cells in vitro, and then a variety of other methods that are essentially, again, addressed to pick up marker changes, such as changes in DNA structure and the ability of cells to repair such damage. Now, one of the important developments that will make a considerable difference in our ability to intervene in a preventive fashion early in the control of exposures, such as occupational exposures, will be that of validating the methods, the short-term methods, so that we can begin to be more and more reliant on those. The advantage of the short-term methods is, of course, that they are very rapid to give us critical answers. They are not necessarily cheap and dirty, quick methods. Some of them are very delicate biological systems that we have to use with professional competence and with caution. But in the proper hands, they constitute very promising approaches to early detection of key effects that may become better and better established as indicators of cancer induction capabilities on the part of the test chemicals. The present cancer policy proposed by OSHA, for example, recommends the consideration of these short-term tests as supporting additional evidence, but not as evidence sufficient by itself in the absence of animal data to demonstrate or to be taken as demonstrating the carcinogenicity of a chemical. In the next decade, I hope we will really be able to pin down this area much more precisely. Now, we, in the last few minutes, come down to the animal test problem. The animal test is still the key tool we have for trying the effect of unknown agents, chemicals, in a biological system, in a mammalian species, which is close enough, as I said before, to the human, to be used as a reliable indicator of the potential effect in the human. There's been, as you know, a considerable amount of discussion on the quality of the test, the criteria for the interpretation of the tests. In the past, there have been, in fact, somewhat different criteria used by different agencies in the government in their interpretation of these findings. A recent effort this past several months has led to what I think is a very useful summary of these criteria, which have been agreed on by scientists and regulatory officers in the key agencies, the Food and Drug Administration, the Environmental Protection Agency, the Consumer Product Safety Commission, and the OSHA, joined in the IRLG, which is the Interagency Regulatory Liaison Group. This group has called on the research agencies in the government, especially the National Cancer Institute and the National Institute of Environmental Health Sciences, to lend them the time and effort and perhaps competence of some scientists, I've been one of them, to work together to prepare these criteria as a review, as a scientific review, as a summary of the state of the art in this field. This lengthy report has been published in the July issue of the Journal of the National Cancer Institute. I've left a few copies of it with the press office, and they were probably going to make some additional copies. If you find them there, fine. If not, please let me know, call me, or just take a note. Is the July issue of the Journal of the National Cancer Institute that has this report, which is entitled Scientific Basis for Identification of Potential Carcinogens and Estimation of Risks. It contains a lengthy, somewhat detailed discussion of all these criteria. Those of you who feel strong enough to go through all this thing and are interested in and have the background to go into the details of these will find what is now, I think, a reasonable agreement recognized in the scientific community. We have had the benefit of very extensive public debate and very extensive contributions from all sectors of science and society through the mechanism of public comment and hearings that the government has in its various agencies. The comment to the OSHA cancer policy is an enormous stack of documentation that has really provided the government with the expert opinion, the documentation, the data offered by a very large range of members of the scientific community. And so have many other such hearings and public comments received by other agencies. So I think this has not been a document put together in splendid isolation by government bureaucrats. This is essentially what I wanted to indicate. The animal model is not an esoteric concept. It is the logical development of current methodologies in science. It's the mainstream of scientific methodology. It is the basis on which we have learned how to cope with most diseases by learning to handle experimental models of them. It would be a tragedy if we were to miss the opportunity to continue to do so in the most effective way, to get early warnings of hazards based on experimental methods that could be used as an efficient basis for protecting people from reproducing the extremely impressive sequence and tragic sequence that Dr. Selikov has illustrated yesterday of continued inaction in the presence of increasing evidence of hazards. I would like now to continue with the presentation of Dr. Ligator, who has to discuss the topic of cancer prevention in this context. Dr. Ligator is professor and director of the Division of Environmental Toxicology at the University of Texas at Galveston. He has previously worked at Brown University and for a number of years at the Food and Drug Administration. His work, particularly in genetic toxicology, is outstanding and well known, and I don't need to spend more time in favor of recommending short time to you. Policy, Dr. Olson.
Thank you very much, Dr. Saffiotti, for your introduction, and thank you, Dr. Epstein, for your introduction.
Thank you. The last panelist on this program is Dr. William McCarville, Director of Environmental Affairs, Corporate Environmental Policy Staff of Monsanto Company. Dr. McCarville is a has a career as a research chemistry. As a research chemist, he has been in the Monsanto company in various positions for many years, and will speak on the topic of wisdom in the workplace. Dr. McCarville.
Thank you, Dr. Saffiotti.